Good afternoon. This session is about the giant chromosomes. And before we start the giant chromosomes, just a small brief look into what we learned in the previous sessions. And the previous session was about the mitotic chromosome and the structure of metaphase chromosome we learned. A typical metaphase chromosome will have a structure like this. It's a typical structure, and these are called the two identical chromatids, and inside this you can see the DNA, and at the center of the metaphase chromosome, you can see the centromere. At the center, there is a centromere where the two chromatids are attached, and this is the region which is having the constitutive heterochromatin. You might have that we discussed in the previous sessions. Constitutive heterochromatin, or the permanent silence, is the DNA sequences which is in permanent silence is found in centromere. So that is known as the center of chromosome, and there are kinetochores, which are the plates on which these spindle fibers attach, and the tip of the chromosome is called telomere, and centromere and telomere are regions where there is constitutive heterochromatin. Yes. Now, today we'll have a look at the giant chromosomes. Giant chromosomes, as the name signifies, giant, they are bigger chromosomes. Apart from the normal chromosomes, the size of this chromosome will be larger. That's why it's called a giant chromosomes. And the giant chromosomes are decondensed during the interphase. And giant chromosomes are actually found in those cells where more protein has to be synthesized. In certain region of the body or certain part of the body of the organisms need more proteins to be produced at specific times. For example, in case of organisms which feed continuously, there are some organisms, in case of human beings, or usually when we talk about the mammals, we occasionally only eat food. But there are other animals like the larval forms of butterflies or insects which continuously feed. When we observe a caterpillar, you might have observed that continuously it will be eating the leaf. It will move from one leaf to another, continuously it will be eating. So that signifies what? There is a need for the salivary enzymes to be synthesized. That is what is called. So that is the significance, or that is the importance of having giant chromosome, or the presence of giant chromosome in some animals. So giant chromosomes are found in those cells where there is intense production of protein or intense production of RNA is taking place. So, and some examples of the giant chromosomes are, as I said, in the salivary gland of the insects, larvae of insects, and the giant chromosomes are found in the oocytes or ovum of the amphibians, or in the oocytes. There also more proteins are synthesized, such as the vitelin. So giant chromosomes are large chromosome too. Where are they found? They are found in cells where intense production of RNA or proteins is taking place. Now, the giant chromosome is of two types. The giant chromosome is of two types. They are the Polythene Chromosome and the lamp brush chromosome. It is of two types, the Polythene chromosomes and the lamp brush chromosome. These are the lamp brush chromosomes, and this is the Polythene chromosomes. We'll look into one by one what a lamp brush chromosome is and what a Polythene chromosome is. First one is lamp brush chromosome. Lamp brush chromosomes are the specialized chromosomes which are found in the oocytes of vertebrates. Vertebrates means those organisms which have a vertebral column. So, lamp brush chromosomes are present in oocytes or egg of vertebrates. In case of birds or reptiles or amphibians, the eggs are reserved place for the storage of proteins or food for the young ones. For example, in case of birds, in case of birds, or the birds will store the food which is required for the developing embryo. So at that place, what is required? Proteins for the baby or the embryo is needed. So they need more proteins. That is why this Lamb-Rush chromosome or this giant chromosome is present in the oocytes. So oocytes are the reserve area for the storage of food for the developing embryo. So more proteins such as vitelline is to be synthesized. So more RNA. So these cells or the oocytes will produce lot of RNA. Why lot of RNA? More proteins is required. So because more DNA is present, the volume or the size of the nucleus also increases. And this actually, the structure is well developed, well studied in the diplotin stage. That is, this chromosome or the Lamb-Rush chromosome or the giant chromosome makes its appearance during the diplotin stage. During the diplotin stage of the meiotic division, the Lamb-Rush chromosome makes its appearance during the diplotin stage of meiotic division. The meiotic division, the diplotin phase കൂടുതലായിട്ട് this you can see the maximum development of this Lamb-Rush chromosomes. And during this phase, the DNA will stretch out its arms. Why giant? It will become larger in size compared to the other chromosome, and it will take a remarkable size or shape which look like a lamb brush. It looks like a shape of a lamb. That's why it's called lamb brush chromosome. It was discovered, or it was first reported by Ruckert in 1892. Ruckert was the first who described the giant chromosome, that is the lamb brush chromosome, in the oocytes. So the Lamb-Rush chromosome are the longest known chromosomes. They are very long. In the length is more. They are the longest. It's the longest chromosome, which a diameter which is less than that of, although the diameter is less than that of the polythene chromosome, and the length varies from 350 μm to 800 μm during the diplotin stage. They are very long compared to the normal chromosome in the body of that individual. For example, in case of amphibians, if the length of normal DNA is 10 μm, in the case in the oocytes, the DNA will be 30 μm or 50 μm in length. That means the size is more when compared to the chromosome in the normal cells. And they occur or exist as bivalents. The chromosomes of the Lamb-Rush chromosome exist as the bivalent. See, this is a Lamb-Rush or a structure, and the chromosome will take a structure similar to that of the Lamb-Rush, which with large number of side loops. Just imagine this is a brush, and that is why it is called a lamb brush. There is a central axis. Here you can see this is the central axis. Central axis is the normal DNA. The normal DNA of that organism will be similar to the central axis. We have a twist and turn like structure for this brush. That is a normal DNA, and that DNA will be present in all the cells of that amphibian. But in the oocytes, there will be some extensions towards the either side. See, there are some extensions to both sides from the normal chromatid, and these extensions show the intensive production of the RNA. And the Lamb-Rush chromosomes are the longest known chromosomes with length of 350 μm to 800 μm, and they occur as pairs. And the two homologous chromosomes of this pair are held together at a common axis. That's called the chiasmata. they are joined at a common axis called, chiasmata. They are joined at a common axis called chiasmata. Chiasmata. With the common central axis. So, from the common central axis, the lateral loops arise or the side loops arise, which help in the intense production of RNA and the proteins. So, the loops represent the lateral extended region of the chromatid. See, this is a paired Lamb-Rush chromosome. This is a paired Lamb-Rush chromosome. Here you can see a normal chromosome will be like this. The zigzag, this black color region is a normal chromosome, will be going like this. This is a normal chromosome. This is the normal chromosome. In this picture it's shown. But these extensions you can see from the normal chromosome, some structures or the finger-like structures arise like this. They are that gives the Lamb-Rush structure, or that increases the DNA content, and that will help in the increased production of proteins. So at the base of each loop there are numerous chromomeres, and the base of this is a region of a single chromosome. So this is a polythene chromosome, and this much part is extended here. This much portion or this part, extended view or an enlarged view of this much region of the polythene, this Lamb-Rush chromosome is shown here. You can see a central axis. This is the central axis. From the central axis arises the loop. These are the loops. Like this it goes. These are the loops which arise from the central axis. And this is an enlarged view of a single axis. This you can see, this is a enlarged view of a single axis. In this enlarged view, what are the structures you can see? Here you can see the chromatin loop and the chromomere. This region is the chromomere. Chromomere or condensed region. Chromomere. Chromomere is the condensed region, or chromomere stain more. Chromomere are the condensed regions which is having tightly coiled loops. This is the chromomere. Just have a look. Normal DNA. Here a condensed loop you can see and an extended loop you can see. And this condensed looping, or when the looping is very close, that is known as a chromomere, and an extended balloon-like structure is found. This is less condensed. So chromomere is more condensed and extended chromatin is less condensed. That means it's much more loose. The size and shape of the loops will vary accordingly based on the amount of proteins which are produced. And these loops are the regions of active gene expression. And this loop, see, this loop or extended chromatin loop are the regions of gene expression. Why? Because it is in a loose form. It is in a expression. Because these are the regions which is decondensed. So that regions are loose, or those regions will favor the formation of RNA, and in effect it helps in the formation or the synthesis of proteins. And so more proteins will be produced. So the majority of chromatin is not in the loops but in the highly condensed chromomeres. Chromomereലാണ് കൂടുതലായിട്ട് chromatin content ഉള്ളത്. പക്ഷെ അവിടെ നിന്ന് എന്താണ് ട്രാൻസ്ക്രിപ്ഷൻ നടക്കുന്നില്ല. But extended loopil നിന്നാണ് more amount of RNA is produced. So hope it's clear. Once more I'll say, giant chromosomes are present in cells where more proteins or RNA has to be produced. Giant chromosome is of two types. They are the Lamb-Rush chromosome and the Polythene chromosome. The Lamb-Rush chromosome was discovered by Fleming and the name was given by Ruckert. Lamb-Rush chromosome, according to the Lamb-Rush chromosome model, there is a central axis. Central axis, you can imagine that as a common DNA itself. And from the central axis arises large number of loops. And these loops help in increased amount of protein synthesis. See, we have always said the finger-like projections or the structures which arises from the chromosome or chromosome will help in the intense production of proteins or intense production of RNA, and in result is intense production of proteins. Where are these Lamb-Rush chromosomes found? They are found in the oocytes where more amount of proteins has to be produced for the developing embryo. Now, this is an extended structure. The Lamb-Rush chromosome will have extended loop-like structure, and this loop-like structure will have a condensed region and an extended loop. Condensed loop, no gene expression takes place. Why? Because the chromosome or the chromatin there will be in a condensed stage, and that is known as chromomere. Chromomere, the chromosome or the chromatin is in a condensed stage, and that is not exposed, or from this condensed stage the gene expression is not present, but from this extended loop more proteins are produced. So that is about the Lamb-Rush chromosome. And the functions of Lamb-Rush chromosome, we said it is involved in the synthesis of RNA and proteins, and each locus codes for the RNA, and the loop is supposed to be the synthesis of large amount of RNA, and they have repeated DNA sequence, and they help in the formation of yolk material or vitelline in the egg. Now, next is polythene chromosomes. Polythene chromosomes are also the giant chromosome, and this polythene chromosome was discovered by Balbiani. Polythene chromosomes were discovered by Balbiani in 1981 in the squash of the salivary cells of Chironomus larva. These polythene chromosomes were first discovered in the squash of the slide preparation of the salivary cells of Chironomus larva. The Chironomus is a larva of a dipteran insect. Chironomus is a larva of, it is the larva of a dipteran insect. Dipteran means those insects which come along with the housefly. Dipteran insect. So dipteran, polythene chromosomes was discovered by Balbiani. Where are these polythene chromosomes found? They are found in the salivary glands of the insect larva. At first it was discovered in the salivary glands of Chironomus larva, the larval form of the dipteran insects. Then they also occur in the rectal epithelium and malpighian tubules, and they reach to a length of almost 200 μm, and it is even then these polythene chromosomes are visible under the normal chromosome or the normal compound microscope which are used in the labs. The normal chromosome with the 10x and 40x lengths. Then the enormous size is because of the duplication of chromosomes, and this polythene chromosomes have 1,000 times more DNA than the normal somatic chromosomes. Polythene chromosomes, the DNA is 1,000 times more than the DNA, more than the normal somatic chromosome, and that's why it is called polythene. Poly means many number. It is 1,000 times more DNA than the normal somatic DNA, so they are called the polythene chromosomes. Then these polythene chromosomes are actually formed due to the repeated divisions, and this longer or the large size of chromosome is due to the presence of long longitudinal strands. Long longitudinal strands undergo repeated division, and that is the reason for the formation of the polythene chromosome. And these polythene chromosomes are formed as a result of the repeated division of chromosome, that is without cytoplasmic division, and that is known as endomitosis. Polythene chromosomes are formed as a by a phenomenon called endomitosis. Endomitosis means what? The chromosome will undergo division, but the cells will not undergo division. So what will happen? The cell will be of normal size, but the chromosome will be larger. Why? Because the cells are not dividing, and that's known as endomitosis. So polythene chromosomes are formed as a result of endomitosis. Endomitosis is a phenomenon in which the chromosome undergo division, but the cells will not divide. Endo, inside the cell, undergoes mitosis within the cell. So polythene chromosomes have the polythene chromosomes contains two types of bands, that is the dark band and the interbands or the light bands. The dark bands constitute 80% of the DNA and the interbands constitute 15% of the DNA. The dark bands are called so because they are stained as dark. They stain dark, and the interband stain light with the nuclear stains, and that's why they are called the dark bands and the light bands. The dark bands contain more DNA and less RNA. And the polythene chromosomes are enlarged at some regions. These giant chromosomes, or this DNA content of the pair of homologous chromosomes, is measured as 2C. So if the DNA content of the normal chromosome is C, the polythene chromosome will have a chromosome content of 1024C. That means 1000 times multiplied the chromosome. That is why the content is 1000 times more than the normal cell or the normal somatic cell in the body of that organism. And these the bands of the polythene chromosome become enlarged and they form a region called chromosome puffs or chromosome puffing. And so this is the larval form. This is the head of this is a chironomous larva, and the chironomous larva salivary gland. If you isolate the salivary gland will be very well functional, because I said the insect larva will be a continuous or a voracious feeder. So the salivary glands will be continuously secreting the salivary enzymes. So they have a very well developed salivary gland, and the salivary gland, this is the salivary gland, this is the salivary gland, and salivary gland you can localize this polythene chromosomes, and the polythene chromosomes will have the light and the dark band. Some parts of the polythene chromosome will be darkly stained and some region will be lightly stained. And in polythene nuclei, the large blocks of the heterochromatin which are near to the centromere. Centromere is the center region of the chromosome of the mitotic chromosome is the centromere. So the large blocks of heterochromatin which are adjacent to the centromere, they are aggregated towards a single point called the chromocenter. See, this is the chromocenter. So in polythene chromosome, the heterochromatin which is near to the centromere are aggregated into a single compact structure called the chromocenter, and the arms of the chromosome will arise out of this chromocenter. So instead of having a structure called centromere, there is a structure called chromocenter in the case of the polythene chromosomes, because the heterochromatin, the large blocks of heterochromatin which are near to the centromeres, they aggregate to a single point. That's why it's called a chromocenter. So in case of polythene chromosome, instead of using the centromere, we use the term chromocenter. And the arms of the chromosome seem to arise from the chromocenter. See, this will be a chromocenter. This is the chromocenter. This is the chromocenter. and from the chromocenter arises the arms of the chromosomes. That's known as the chromocenter. The arms of the chromosome arise from the chromocenter. So of the 4 pairs of chromosomes, the largest ones, that is chromosome number 2 and 3, have centrally located centromeres, and therefore the right and left arms appear to be equal in size. Then the chromosome number 4 has a short arm. You can see the right arm of chromosome 3, the left arm of chromosome 3. It will be almost of same size. Then you can see here the chromosome number 4. The chromosome number 4 appears as a short but projecting from the chromocenter. The chromosome number 4, see, the chromosome number 4 will be shorter in length. But chromosome number 2 and chromosome number 3 will be of normal length only. And these chromosomes are visible under this microscopes and are usually the stains which are used to observe this are acetocarmine or Fulgen stain. And this 80% of the DNA is present in the bands and 15% are found in the interbands. And why? There are estimated that the polythene chromosomes are very much active, or they are the active sites for the synthesis of proteins or RNA. And this is the entire chromosome of the Drosophila. You can see here the chromocenter, and this is a long chromosome. Because there is no division of the cell, the chromosome size will increase enormously. The chromosome length or the chromosome size increases because there is no division of cell. But polythene chromosomes, during particular stages of the larval development, these chromosomes are puffed out. This is called chromosome puffing. These structures are called chromosome puffing. Chromosome puffing. So the polythene chromosomes have at certain region of the chromosome, polythene chromosome, some regions are enlarged, and these enlarged regions are called chromosome puffs, and this phenomena is called chromosome puffing. Why this chromosome puffing? The chromosome puffs means from the central axis, the active site of gene expression. Some extensions of the chromosomes are found from the central axis, and that is known as chromosome puff. And this chromosome puffs are the regions which of intense production of proteins and RNA. And at these sites, the active gene expression is found. Active gene expression means what? More proteins are produced. Why more proteins? More salivary enzymes has to be produced. Why more salivary enzymes? Because the organism is continuously eating food. That is the chromosome puff. And this in the bands or in the interbands. Here you can see in this bands there are some dark bands and light bands. And in the dark bands there is no transcription. In the dark bands there are dark bands. Here you can see the dark bands. Under the microscope, certain portions will be stained dark and certain portion will be lightly stained. And this dark stained regions are condensed. The dark stained regions are condensed, or they will be having heterochromatinized DNA. The dark region means they have heterochromatinized DNA or condensed DNA. Dark region is the DNA is condensed. So, because the DNA is condensed, no transcription. No transcription from this dark bands. But from this, when the protein synthesis is required, the DNA of the bands will undergo decondensation. Hmm? So in the bands, the DNA will be in the condensed form and there will be no transcription. But when there is a need for the synthesis of proteins, the chromosome will undergo puffing. Puffing means decondensation. Puffing means decondensation. Decondensation. Puffing means decondensation. So when protein synthesis is required, the DNA of the bands will form puffs. Puff means what? Puffing means decondensation of the DNA takes place, and as a result more RNA will be synthesized, and some of these puffs will become enlarged, and these enlarged puffs are called Balbiani rings because it was discovered by Balbiani. It is called Balbiani rings. Balbiani rings. Chromosome Balbiani rings. So puff formation is induced by the hormone. This how this puffing is formed. So this puff formation or the extensions of this chromosomes are formed as a result of the influence of certain hormones. Which hormone? Let's see which hormone is it. So this puffing or the formation of chromosomes is with the help of a hormone. Chromosome puffing is happening in the presence of a hormone. Happens in the presence of a hormone in insects. Which hormone is it? Ecdysone. The hormone which is involved is ecdysone, and ecdysone is called the molting hormone. Why molting hormone? When an insect metamorphoses from one stage to another, a hormone is introduced in the body of this insect, and that's called the molting hormone. So, what during when an insect larva undergo metamorphosis from one stage to another, the hormone produced in its body is called the ecdysone or the molting hormone. So the molting hormone trigger the formation or molting hormone helps in the formation of the chromosome puffs in the insect larva. Why the insect will undergo metamorphosis from one stage to another. So when you inject or when you apply this ecdysone onto the insect larva prior to molting, that will result in the pre-molting or the molting will be preponed. The hormone ecdysone, if we apply to this larva, it will molt before its normal time. That is known as that is the significance of chromosome puffing. So, polythene chromosome is a giant chromosome found in the salivary gland of the insect larva of insects. It was first discovered by Balbiani. Then the polythene chromosomes are the polythene chromosomes undergo repeated division, and the repeated division which is happening in this repeated division which happens in but this polythene chromosome is called endomitosis. As a result of endomitosis, the chromosome will increase in number, but the cell will not divide. That is endomitosis. Endomitosis means chromosome doubling without nuclear division. And This chromosome, which is of longer length, will be attached to the chromocenter, and chromocenter will be having heterochromatinized region. It's a heterochromatinized region, and there the chromosome is in a decondensed. It is in a condensed state. So when you observe this chromosome under the microscope, you can see the light and the dark bands, and the dark bands or the bands on the chromosomes are regions which is condensed, are those regions which are having condensed chromosomes. So when there is a need for synthesis of proteins, like when there is a need for the synthesis of the salivary enzymes, chromosome puffing is formed, or chromosome puffs are formed. What are the chromosome puffs? They are the enlargement or extensions of these chromosome of this bands or this Balbiani. They are called the Balbiani rings. So Balbiani rings are the region where the puffs or chromosome puff is formed on the chromosome. They are transcriptionally active region. Transcriptionally active means more RNA is produced and more proteins are produced from these region. And in case of insects, this chromosome puffing is initiated, or chromosome puffing is aided with the help of the hormone called ecdysone. Ecdysone is the molting hormone